I think I turned on closed captioning. So today is our 60th Brainstorm Live, if I counted them right.
Wow.
And yeah, right. So if you've been to all 60, I guess we should get some kind of medal or something to present to you. But today we have Dr. Matt Carter talking about sleep and hunger, and I'll let him introduce himself.
Great. Thank you, Sasha. And thanks to everybody for being here. This sounds like a wonderful group. Sasha was telling me a little bit about it, and sounds like a really great way to kind of translate some recent neuroscience and brain fact findings into the profession. So it sounds really good. And Sasha said that people had gotten some benefit out of a book that I helped write called Guide to Research Techniques in Neuroscience. And I remember we wrote that years ago because we couldn't keep track of all the techniques ourselves, and so I'm glad that it's always nice to hear that resource is useful for some people. But yeah, it's a pleasure to talk with you today. I'm a professor at Williams College in Western Massachusetts. I'm actually on a trip right now, so I'm calling from Las Vegas. Not it's not the kind of Las Vegas that comes to mind when you hear that term. I'm actually in a conference center that's about 15 minutes from the Strip. But yeah, it's a pleasure to talk to you. My lab studies sleep and hunger, and there are some projects in which we work on both of those motivational behaviors and at once. And that's what I thought I'd talk about this morning, is the context of how hunger affects sleep. And in addition to researching sleep and hunger in my lab, I use mice as a model organism in my lab. I teach classes in physiology and neuroscience at Williams, and I think my main way of doing something of service to the Williams community is to have seminars about sleep. And so I'm often asked to give talks about sleep to undergraduates and to athletes. Actually, a lot of the coaches ask me to go give talks about the importance of sleep to their teams, and so I'm a big proponent in getting a good night's sleep. And there's another sleep scientist named Matthew Walker who wrote a book called Why We Sleep, you may have heard of. It's a really popular book, and I really love it. And sometimes people confuse me with Matthew Walker because we have the same first name, and I'm a big proponent on sleep, but he wrote the book, not me. But I am a big proponent on getting a good night's sleep. And so what I thought I'd do is just share some of the results of a recent project that my lab worked on, and talk for a little while about some of the scientific results that we found, and then help have a conversation or answer any questions that people have. But I think for a group like this, I mean, I think that the benefits of getting a good night's sleep are hopefully very clear to everyone. You know, I've studied sleep now for about 20 years. My PhD research was about sleep, and there's tens of thousands of studies about sleep, and I've never seen one that shows that it's beneficial to not get enough sleep in any way. Everything improves with getting enough sleep, and I think that actually sleep deprivation and poor sleep hygiene is one of the big epidemics in our society right now. Is just the fact that I don't think most people fully realize that if they committed to getting enough sleep every night, that so many aspects of their health and their well-being would improve. But one of the things I study in my lab is what are the factors that lead people to not get enough sleep. And I'll just share my screen here and share some of the recent project that I've worked on. Let me go here. Great. Can you all see this slide? Yeah, so this is a scientific project that my lab worked on, and again, I work with mice, and so I think a great conversation topic is how this translates into humans. The nice thing about working with mice is that many of the same brain regions and structures that are in humans are also in mouse brains. Mice are obviously not humans. They're, especially in their cultural lives and their familiar lives. But from the point of view of survival systems, and the systems that keep us alive as animals, mice and humans actually have a lot in common, and just about all of the same populations of neurons and groups of neurons in the brain that keep humans alive, you can find in about the same locations in the brain in the mouse, and how they're connected to each other. And so I think that mice, there are obviously limitations to studying mice as a model for humans, but I think when it comes to things like hunger and sleep, mice are actually a really great model to study humans. And one of the things that I encourage my students to think about, and that I've gotten a lot of meaning of myself, is just what are we as animals evolved to do? How have we evolved for maximal health? And when I teach my physiology class, we talk about the factors that are necessary for survival. And so this is kind of a day one activity I would do in my physiology class, is to brainstorm a list of what are all the factors that an animal needs to survive, and how does an animal what are the factors that an animal needs to maintain homeostasis for? And so these are some items that would come on a list. Is obviously animals need oxygen, sleep, we need to maintain an optimal pH, water, optimal pressure is something that a lot of people don't think about. Actually, in the news, I feel really bad for the submersible story, but I think that's an example of how we all need to maintain an optimal pressure. There's a lot of animals that actually do have to think about this on a regular basis. We humans often do not, but that's an unfortunate news story. Yeah, we need to defend ourselves from predators. We need to maintain an optimal temperature. We need to have a way of removing wastes and obviously consuming enough nutrients in our food. And so we humans don't often think about our daily lives in terms of all of these things, but obviously part of our brain, our unconscious brain, is constantly trying to maintain optimal levels of these different homeostatic factors, whether we're conscious to it or not. And when I look at this list as a neuroscientist, I recognize that some of these factors, an animal has to change their behavior in order to achieve. So in red here, seeking food, sleep, water, and optimal temperature, many times we have to change our behavior and our motivational state in order to seek these things, as opposed to oxygen, which our autonomic nervous system usually generates for us. And the interesting thing about some of these is that we can pursue and be motivated to seek some of these at the same time. So, for example, we often have water and food together at the same time. Sometimes the food and the water that we consume is at an optimal temperature. I like drinking coffee and hot chocolate on a cold day, and I like drinking ice water or lemonade on a hot day. So a lot of these factors we can seek, and they're not mutually exclusive. But a couple of these are mutually exclusive. And this is a project that my lab has recently been working on, is that it's impossible to eat and to sleep at the same time. There was one time I was so tired that I fell asleep while eating, like a particular embarrassing moment of my life, and I woke up realizing that I'd fallen asleep. But a human and a mouse can't eat and sleep at the same time, and so basically wanted to know, how does the brain balance both of these behaviors, and how does the brain decide to commit to one of these behaviors more than another? So really, we're asking, how does the homeostatic drive for food affect sleep quality and sleep quantity? And as you may know, sleep in humans is measured using a technique called electroencephalography. This is a technique where electrodes are put on the outside of the scalp in a non-invasive way. There's no surgery needed for this. And what each little individual electrode is doing is measuring the neurons that are right under the scalp. And usually it's measuring millions of neurons neurons at a time. And this is useful because the pattern and the synchronization of how those neurons are firing at once can be useful for measuring the quality of sleep states. And so in a human, you may have seen a picture, an image of a human wearing an EEG setup, but we can do this in the lab with mice. We do a little surgery on the mouse where we actually install little electrodes on the scalp of the mouse. And we do this during a surgery so the mouse doesn't feel any pain. The mouse isn't under distress. And actually, before the session was started, I was talking with Sasha and Eric just about the ethical training that we go through in order to do this, and the ways that we minimize distress in the mice. But when we record using EEG and another technique called electromyography, which is measuring the muscles of the mouse, we can basically determine 3 states in the mouse: is whether the mouse is awake, and or discriminating between 2 sleep states: non-REM sleep, non-rapid eye movement sleep, and rapid eye movement sleep. And I'm showing you what these traces look like here, mostly to show you that they look differently from each other. And in my lab and in sleep labs, people can be trained to look at these waves that are recorded from these electrodes on the scalp, and also the motor activity, and discriminate between these 3 different states. And so if the mouse is motionless, just by looking at the mouse, it's hard to tell whether it's asleep. Maybe it's awake, it's just not moving. And then if it is asleep, what does that sleep look like? What is it, non-REM sleep or REM sleep? But this is a way for us to be able to look at exactly what this sleep looks like. And so one of the first things we did in the lab was just to ask what happens if a mouse is hungry? How does that affect the sleep quality and the sleep quantity in the mouse? And this is actually a reason I like using mice as well, is because you can study the effects of food deprivation in people, but obviously people's lives are very complicated. There's so many confounding factors that happens in human studies. And so the advantage of using mice in this case, again, mice aren't people, but we can really reduce the confounding environment for the mouse and really study what specifically is the effect of being hungry on sleep states. And so here's a graph that I'll just sort of walk through. Is on the x-axis is the different sleep states, so wake, non-REM sleep, and REM sleep. And we food deprived mice for about 16 hours. And so they're hungry, and then when we measured their sleep states during the period when they would normally go to sleep, the light bars are mice that are ad libitum fed, so they had constant access to food. And then this is 24-hour food dep. So these mice have been food deprived for 24 hours, and you can see basically they increase the amount of time that they're awake, and they decrease the amount of time that they're in non-rapid eye movement sleep. And so presumably this is because they're hungry, they're looking for food, and they're in a state where they're more motivated to look for food and to consume food, and so the quantity of their non-REM sleep goes down. But if there's something that we found that was also interesting, it's that when they did go to sleep, the quality of their sleep also decreased. And so usually when people think of a good night's sleep, they think only of quantity. They think, for humans, we need about 6 to 8 hours of sleep per night. But even within those 6 to 8 hours of sleep, some nights of sleep are better than others. And one way that we can measure the quality of sleep is by looking at actually the power spectra of those electroencephalography waves. And so without getting into the details here, it's basically we can look in those waves, and we could look at the frequency of the waves. And there's a specific kind of frequency called the delta frequency, which is on this bar on the right. And it's basically what is the frequency with which we see these kinds of upward deflections and downward deflections of the EEG. And the delta frequency corresponds with the quality of non-rapid eye movement sleep. And so what we found is in an animal that's hungry, the delta power of that frequency range of non-rapid eye movement sleep decreases. And so not only are the animals getting less sleep, but the quality of sleep when they do get sleep is going way down. And for humans, if you've ever talked to somebody, or you probably experienced this yourself, sometimes they get a full night of sleep in terms of their quantity, but they sometimes sometimes say, wow, I don't understand. I still feel really tired and groggy, even though I got 6 to 8 hours of sleep. And the chances are that the quality of sleep may not have been great, even though they got the right quantity. So food deprivation decreases non-REM sleep quality. The last thing I wanted to say about quality is something that most people don't know about sleep, actually, which is that usually during the night, a person and a mouse will have these very brief periods of waking up, and we sleep scientists call this micro awakenings. And a micro awakening, this is an example shown here. So here's an EEG trace on the top and a motor trace of the muscles at the bottom, and a little scale bar here. So this was recorded from a mouse, and if you look at this 5-second period right here marked by the arrows, here's a mouse that was asleep in non-REM sleep. And then just for a few seconds, you can see a change in the EEG and the EMG. And this mouse woke up just for a few seconds and went right back to sleep. And this happens in humans also. In fact, this happened in you last night. So last night, you underwent several micro awakening events, and you actually woke up for a few seconds sporadically throughout the night. And you have no memory of this. This isn't something that we're conscious of. In fact, your eyelids might not even open. But we all go through these periods of micro awakenings. And what scientists have shown is that the number of micro awakenings that a person has correlates with a loss of quality of their sleep. So if you have a lot more of these little micro awakening events, then the quality of your sleep goes down. And what causes these micro awakenings? The frequency with which a person will have micro awakenings goes up if you're more stressed out, if you have had sugars in your diet or alcohol in your diet, especially just before going to bed. Unfortunately, the frequency of micro awakenings goes up the older that you are. So somebody who's older will naturally have more micro awakenings than somebody who's younger. All of these leads to micro awakenings, and micro awakenings fragment sleep and make the quality of sleep much worse. And so in looking at this in terms of being hungry, what we found was that mice that were hungry had much more, about twice as many micro awakenings as mice that were not hungry. And so going into the inactive period where mice normally go to sleep, if they were hungry when they went to sleep, the frequency of the micro awakenings went up, which leads to a worse quality of sleep. And having a worse quality of sleep would lead to effects in learning and memory and cognition the next day. So the take-home point of some of these initial mouse studies we did was that food deprivation decreases sleep quantity and quality. And I think like one obvious impact of this and application of this is that people who might not have enough food or are facing conditions of food insecurity, going to bed hungry actually changes the ability to fall asleep and then the quality of the sleep when somebody is actually sleeping. And so continuing this on, I'm a neuroscientist. And so part of me also wanted to know, like, what are the underlying neural mechanisms of this? And so I thought I would share this to some degree with this group, and would be happy to take this any way people want. But there's a specific kind of neuron in the brain that makes a protein called a Goody-related peptide, and everybody in the field calls this AGRP for short. And so what I'm showing you here is a little cartoon of a sagittal diagram of a mouse brain. So the eyes would be over here, and the back of the head would be back over here on the right side of the slide. Here's the mouse cerebellum, the mouse olfactory bulb over here, but down at the very, very bottom of the brain, kind of in the center of the brain at the very, very bottom, it lives these neurons, which are called AGRP neurons. And AGRP neurons are very well known for promoting hunger. They're the primary neurons in the brain that detect what you've eaten and the nutrients that are in your bloodstream. And the longer that a person or a mouse goes without eating, the higher the activity of AGRP neurons. And so these neurons are very, very, very active the longer that an animal has been without food. And so these neurons are well known to regulate feeding behavior. Here's a picture of them, by the way, if you want to know. It's like, so these little red dots here are individual cells. These are the neurons. So they're expressing a red fluorescent protein, and all of the other neurons are expressing a blue fluorescent protein. So here's a bunch of blue neurons, and they're in the very center of the brain surrounding a ventricle, which contains cerebral spinal fluid. And so this is the very center of the mouse's brain. And then on the just to the left and just to the right is where the AGRP neurons live. A mouse has about 10,000 AGRP neurons, and a human has many more than that. And so these are the neurons that make you feel hungry. So the next time you're feeling really hungry, you can know it's your AGRP neurons that are making you feel that way, and they're firing like crazy. So we thought, what would happen if we turned these neurons on artificially? So if a mouse had plenty of food and it was well fed, what would happen if we turned on AGRP neurons, and how would that affect sleep quantity and sleep quality? And the way we did this, I won't get into the weeds of the technique, but there's a technology called optogenetics, which allows us to basically be able to turn these neurons on whenever we want to in a mouse's brain by making them sensitive to light. And then we can do a surgery where we install a tiny fiber optic cable right above the AGRP neurons, and we can shine light through the cable while the mice are freely moving around or doing whatever they're doing, and we can artificially turn these AGRP neurons on whenever we want to. And so this is a top-down, on the right side of the slide, here's a top-down view of the mouse's skull, and this little box here with this 2x3 yellow dots, this is the EEG implant that we put on. And then we also put on a little optical fiber right next to that can stimulate the AGRP neurons. And so we can basically make mice hungry no matter what they've had to eat. And so what we did was we artificially turned these neurons on. And this is a graph basically showing that the blue bars have the light sensitive protein, and the gray bars are animals that don't. So this CHR2 cherry over here, this is the name of the molecule that makes these neurons sensitive to light, whereas TD tomato is a control vector that we use that is not sensitive to light. So really all the comparisons that I'm going to show you are between gray and blue bars, and the blue bars have neurons that where the neurons are sensitive to light. And when we shine the lights of different frequencies at 1 hertz, there's not a difference between the control mice and the mice that have their AGRP neurons stimulated. But if we flash the light more quickly and we increase the stimulation of AGRP neurons, then what this graph is showing you is that we can make the mice consume more food. So we're making the mice more hungry, essentially, by turning on their AGRP neurons. So then the question is, what does that do to sleep? And so the finding that we had was that if we artificially turn on these AGRP neurons that make mice hungry, that also increases wakefulness and decreases non-REM sleep, which makes sense because this is very similar to what would happen if we food deprived a mouse. And so whether we food deprive a mouse or we artificially make the mice hungry, wakefulness goes up and the quantity of time spent in non-REM sleep decreases. So this is the effect of stimulating these neurons on the quantity of sleep. What about the quality? So if we look at the sleep quality, again, here's a power spectrum, and we looked at the delta power during non-REM sleep, and similar to food depriving animals, the quality of the sleep as measured by the power spectra of this delta frequency also decreases. So by turning on these neurons artificially, we can reduce the quantity and the quality of sleep. And then this final graph over here on the right is showing you that the number of micro awakenings, the number of times they have these very brief periods of waking up while they're sleeping, it also doubles a lot. So just by turning on this one little population of neurons, it basically will decrease the quality and the quantity of sleep. And this is I wanted to show you basically here that this is a a trace. Again, this top trace is a control animal in which we didn't stimulate the neurons. And so what this is showing you is on the left here is the animal's awake. And then right here at this dotted line is when it enters non-rapid eye movement sleep, and it goes through non-rapid eye movement sleep. When we turn on the light, this top trace, again, is from an animal where it's insensitive to light. So there's no change, and the animal stays in non-rapid eye movement sleep, and everything is nice and fluid, and it has a nice fluid sleep signature. The bottom trace, though, these are the neurons, the animals in which the AGRP neurons are sensitive to light. So when the animal enters non-REM sleep, and then we turn on the light to stimulate the AGRP neurons, you can see here these little brief moments where the sleep is fragmented. And even though the mouse is in non-REM sleep, it increases the chances that there's going to be one of these micro-awakening episodes where the mouse wakes up, even just for a few seconds when these neurons are active. This also, what we found was that it increases the transitions from wakefulness to from non-REM sleep to wakefulness. So, for example, this graph on the right is basically showing that when we turn on AGRP neurons, there's a higher likelihood that it's going to transition from non-REM sleep into wakefulness and will wake up in a way that's much greater than if these animals didn't have their AGRP neurons artificially stimulated. So the conclusion of this is that if we artificially turn on these AGRP neurons and make mice a little hungrier than they would have been before, that also reduces the quantity and the quality of non-REM sleep. So let me show you one other kind of experiment that we did. We basically wanted to know if these neurons, if the activity in these neurons was necessary for the effects of food deprivation on sleep quality. So basically what this means is, if we food deprived mice, but then we artificially inhibited these neurons and stopped them from firing, would that rescue the effects of food deprivation? And so we did this using a method called chemogenetics. And I won't get into the methodology of this, but this is a way basically where we can inhibit AGRP neurons artificially in a mouse that's moving and free to move around its cage. We can just artificially take these neurons and shut them off so that whatever would cause them to be active normally won't make them active anymore. We're just artificially gonna turn them off. And so what we found was that actually, if we inhibited AGRP neurons, it rescued the effect of non-REM sleep in food-deprived animals. So on the left here is a comparison between control animals, and in orange is the animals in which we inhibited the AGRP neurons. And in an animal that is not food deprived, there's no difference between having the AGRP neurons normal or turning them off. These animals are full, they're well fed, and their sleep is exactly the same. But if we food deprived animals, so we made the animals really hungry, but then we inhibited AGRP neurons, it actually rescued the effects of the food deprivation. So instead of wakefulness going up, the amount of wakefulness went down, and the amount of non-REM sleep went up. And so what this is showing essentially is that these AGRP neurons that trigger hunger in animals are responsible for hungry animals waking up much more than they should be when animals are hungry. And also, when we inhibited AGRP neurons, it reduced the number of micro-awakenings in food-deprived animals. So here's animals that are not food-deprived at all. There's no difference between normal AGRP neuron activity and inhibited AGRP neuron activity. But in food-deprived animals, inhibiting AGRP neurons rescues the effect of food deprivation. So it's basically saying that in a hungry animal, if we inhibit AGRP neurons, that will rescue the effect. And again, here's the power spectra of AGRP neurons. And the key thing is to show that if we inhibit AGRP neurons, this rescues the quality of non-REM sleep and makes non-REM sleep just as good as it would be before. So inhibition of AGRP neurons rescues the sleep quality in food-deprived animals. So the last thing I just want to show you is I've shown you what happens if we stimulate AGRP neurons. I've shown you what happens if we inhibit AGRP neurons. And the last thing I just want to show you is what happens if we just measure what AGRP neurons are doing when an animal goes to sleep. And this is, I think, the most interesting part of everything. So it's basically saying, let's say that you're a mouse or a human and you decide to go to sleep. What changes in your AGRP neurons that make you feel hungry? Do they just, if you're hungry and your AGRP neurons are increasing their activity and you decide to go to sleep and you fall asleep, are your AGRP neurons still just as active, firing in your brain, trying to get you to eat, or does something change in AGRP neuron activity once you go to sleep? And so the way we did this is basically to do a technique called fiber photometry, where we make neurons actually glow more the more that they're active. And so we put in a molecule that glows green, but it only glows green when the neurons are active. So by measuring the amount of light that comes out of these neurons, we can actually determine how active the neurons were. And so we used a method, called fiber photometry to do this, and did this similar to the other experiments. But here's what I'd like to show you, which I think is very interesting. And so here's an animal in which we're looking at the activity of these AGRP neurons. And on the left, it shows you what happens when the animal's awake. And then this dotted red line shows the difference between the animal that's awake going into non-REM sleep. And so this dotted red line is sort of the border between the animal being awake and going into sleep. And this bottom trace down here, this purple and green line, the purple line is a control line that basically is just a control to make sure that there's not a lot of artifacts or a problem with noise in the recording. But the green trace represents the level of activity in AGRP neurons. So what I hope you can see is that the green trace, right when the animal enters non-REM sleep, this trace starts to decrease and goes down and then stays down. And what this is showing us is that when an animal goes to sleep, the part of the brain that makes the animal feel hungry turns itself off automatically. So it's kind of like the part of the brain that's trying to get the animal to go eat. Once the animal goes into sleep, it shuts itself off and it's basically inhibited. And I'll show you this. This is a summary trace from many animals. So this is an average of what all the traces look like. But this top trace is showing what happens to AGRP neurons when an animal goes from wake to non-REM sleep. And so this dotted line is showing you the transition. And so this is a summary of essentially what I just showed you, is that AGRP neurons shut themselves off when you go to sleep. And then this bottom trace, I think, is just as interesting. What this is showing you is that when an animal is in sleep and then wakes up, all of a sudden the hunger neurons turn themselves back on again. And so it's kind of like the brain is committing to something. It's basically committing to, okay, it's time to go to sleep. So we're going to put all of our effort into going to sleep. So we're going to take the other parts of the brain that are trying to get you to eat, and we'll shut it off. And then as soon as the animal wakes up, the brain basically will turn on that hunger drive right again. And so what we think is happening is that in an animal that's really hungry, the hunger, it shuts itself off, but that the hunger actually still is kind of interfering with the quality of sleep by still trying to drive a little bit of hunger, even though these neurons are, some part of the brain is trying to shut them off. And I'll end with one last little thing that I think is interesting, is that, you know, if you remember, so that these micro awakenings, these are times where an animal will wake up for just a few seconds each. What we found was that there's a slight burst of activity that precedes micro awakenings in hungry mice. So basically what this means is that if an animal is hungry and goes to sleep, it's basically abnormal fluctuations in these hunger neurons that are causing the animals to wake up, and that you can predict when a micro awakening is going to happen because of these bursts in AGRP neuron activity. And so again, if I was just going to sum all of this up, what I would say is that when these mice commit to going to sleep, so they go from wakefulness into a sleep state, the AGRP neurons shut themselves off, which makes sense, so that the animal isn't motivated to consume food. But in an animal that's really hungry, the AGRP neuron activity is so high that it can still interrupt sleep, and that the firing of these AGRP neurons can still interrupt sleep. So the overall conclusion from the study that we did was that AGRP neuron activity increases wakefulness and decreases the quantity and quality of sleep. And if an animal is well fed going to bed, then AGRP neurons aren't firing anyway, because there's not the animals have enough food and AGRP activity is low. However, if an animal hasn't gotten enough food, then AGRP neuron activity is high, and the AGRP neuron, when those neurons are active, it interrupts sleep, both the quantity of sleep and the quality of sleep. And so this was a paper that we had published in Current Biology a few years ago, and we've been following it up with some experiments ever since. I'll just end also just by saying that a number of undergraduates at Williams worked on this, and it was funded by the NIH and by the NSF. And so I always like to give credit where credit due, and a lot of the students who worked on this are now in great graduate programs or medical programs myself. Themselves. But I will stop the screen sharing and basically just say that what I conclude from all of this is obviously, again, I'll end the way I began, that these are experiments that were done in mice. What I take from this is basically the fact that as humans, it just seems automatic that at night that we should go to sleep, and in the morning we should wake up. And we have a circadian drive that tries to maintain that. But if we have competing demands, the competing demands can interrupt the quantity of sleep and the quality of sleep in a way that we don't even know about. My guess is almost nobody in the world knows that they have AGRP neurons. This is something that's very specific to my line of science. And so most people they're not walking around wondering what their AGRP neurons are doing. And yet these neurons are trying to compete with sleep, trying to get the motivational factor for a drive that they need. And just like AGRP neurons regulate hunger, there are other neurons in the brain that are trying to make you drink water when you're thirsty. There's other neurons in the brain that are trying to get you to be to potentially deal with a threat when you're stressed out. And all of these different parts of the brain are competing with each other to try to get you to change your behavior. And so the take-home point that I have when I try to get a good night's sleep is basically to try to think of what are the different aspects of survival or evolutionary survival, and how can I get them to not compete with that sense of going to sleep. And so basically what this means is making sure I don't go to sleep hungry, thirsty. I try to reduce the amount of stress that I have when I go to sleep, and just try to diminish anything that's going to interrupt not just the quantity of sleep, but the quality of sleep that I have. So I'll stop there, and I'd be happy to answer any questions. And I see that there's some comments in the chat, but I haven't seen what they are yet. And I'd be very happy to answer anything or talk about anything that would be useful to this group. So thank you very much.
Thank you. I think our very first question was from Marissa. I'll read it aloud, but Marissa, if you want to add to it now that you've seen more. Question she wrote in the chat was, what about when people sleepwalk or other activities? Yeah, talking about sleeping and eating simultaneously, I guess.
Yeah, no, that's a very good question. So sleepwalking and moving when you're asleep is it's a rare experience, only experienced by some people, and it's a dysregulation, especially of rapid eye movement sleep. So usually during sleep, Obviously, humans, we have a laying down posture that we go into. And then during rapid eye movement sleep, normally all of the muscles of the body are inhibited, except for the eyes, actually, which is why it's called rapid eye movement sleep. But sleepwalking is it's a rare experience that happens. And you're right that in people who sleepwalk, it actually is possible to go eat while sleeping, but I would say that that's an exception to the rule and not the rule itself. And people who are sleepwalking don't normally know that they're eating. In fact, they're not in a full conscious state where they know what they're doing. So it's not exactly the same as eating and sleeping at the same time, even though it literally might be. But it's a good question.
The next one I'm seeing is, do micro awakenings inform parasomnias, or is it the other way around?
Yeah, it's kind of both, actually. I would say there's parasomnias are often correlated hand in hand, and so micro awakenings can be causal for some conditions, and then also those conditions can also lead to more micro awakenings. And so I would say that they're both true. People have done a lot of experiments in rodents with micro awakenings, and just like sleep deprivation, they're correlated. No one has ever shown a benefit to micro awakenings. I'll put it that way. Micro awakenings are, I would just go so far as to say that they're not good. They're like the more micro awakenings and the higher the frequency, any kind of measure of behavior when the animal is awake, such as like learning and memory tasks or creativity tasks or things like that, goes down with the increase in micro awakenings. And the symptoms of diseases, whether it's a sleep disease or even not a sleep disorder, but some other kind of disorder, the symptoms of those goes up with the frequency of micro awakenings. So they go hand in hand.
The next one I'm seeing is asking if there's an effect on sleep from overeating or being too full beforehand.
Yeah, that's a great question too. It depends on the nutrients some of the time. So the not all foods are equal in terms of what they do to sleep quality and sleep quantity. And it seems like the bad guy is carbohydrates and sugars. Like that's the one that if you had to select a nutrient not to consume before going to sleep, it's sugars. And the reason for this is that there are specific populations of neurons in the brain that are actually geared towards waking you up. It seems like one of their big jobs is to create alertness and a feeling of energy and vigor. And they're very sensitive to carbohydrates. And if you have an increase in blood sugar, those neurons fire more. And so basically what that means is that if you have carbohydrates like a cupcake or something right before going to bed, it increases the chance that those neurons are going to be active and actively trying to get you to wake up. Another interesting thing is alcohol, which is funny because alcohol is a depressant. And so, in fact, some people have a drink or something to help them fall asleep, which is ironic because, a glass of wine or a beer or something, it will make a person feel sleepy and help them fall asleep. But then once you fall asleep, alcohol is metabolized by the liver into carbohydrates. And so it might help you fall asleep, but somebody who might, drink a glass of wine or something or have a cocktail or something and then fall asleep, 2 hours later, the quality of their sleep is going to go down, and it increases the chance that they might wake up after that. But other states of being full, I mean, I think everybody knows, I call it the post-Thanksgiving effect. It's just like you have a big meal or some like a big hearty meal, and you feel really tired, and your parasympathetic nervous system is engaged, the rest and digest part of your nervous system, and that can definitely help you fall asleep. I think the only thing to watch out for is the carbohydrates and everything else. But proteins and different kinds of fats typically don't wake a person up. It's really the sugars.
The next one I'm seeing from Marissa is a little longer. The first part of it is asking if constant deprivation is different from occasional deprivation.
Yeah, that's a great question. And I see, yeah, part of the question is about fasting and dieting as well. So this is a really great question. And for sure, constant deprivation does have a different effect. So this is actually something I think is really interesting about the nervous system, is that when perturbed in a very acute way, like a very short-term way, the body responds in a certain way. But if you constantly deprive it of something, then the body adapts in more of a long-term way. And I myself have tried intermittent fasting. I don't know if anybody else on this Zoom call has, but there's lots of amazing effects actually with intermittent fasting. And I'm not a medical doctor, so I should be very clear that I'm not giving anybody advice or anything like that too. But the amazing thing is there was one time I tried fasting for 5 days actually. It was a very interesting experience because the first day I was hungry and I did have difficulty falling asleep. And because I had done this study with mice, I think I know why I had difficulty falling asleep. And I just really wanted to eat something, but I couldn't. The thing that was a big surprise to me is that the second and the third day, I felt just fine. I wasn't hungry anymore. I was actually just shocked how my feelings of hunger subsided. And people are looking at the mechanisms of what happens 24 hours later, 48 hours later, when an animal's in one of these constant states. And people don't know the full details of it. But evolutionarily, it makes sense, actually. If you think about an animal in nature, not a human in modern society, but an animal that has to forage for food, you would imagine that over the short term of 6 hours, 12 hours, 18 hours, the drive to go eat would be very substantial to go find food. But if there's something that's wrong, if an animal goes for 24 hours or 48 hours without food, then the other mechanisms kick in order to get the animal to have long-term ability to survive whatever situation it's in. And so there are definitely differences between constant deprivation of things rather than an occasional acute effect. So it's a very good point.
I also see that there's another question about distinction between voluntary or non-voluntary.
Yeah, so this is an interesting question as well, because from the point of view of your AGRP neurons, whenever your AGRP neurons are active, or at least in mice, I could say mice and rats, they always motivate an animal to consume food. And I think this is maybe one of the big differences between rodents and humans, is that a rodent never voluntarily food deprives itself. A rodent, when a rodent is feeling hungry, the rodent's motivated to consume food. Whereas we as humans, we know when food is available and when it's not. We know when we are selectively depriving ourselves of food. Like my decision to fast for 5 days was a voluntary decision on my part. And we also just know what it's like to, maybe if you're hungry at 3 p.m., but then you tell yourself, well, I am hungry, I do have food here, but I'm going to wait till dinner. And so we can make those decisions that rodents don't make. However, there are top-down neural systems. And what I mean by top-down is that the more executive parts of the brain can actually send axonal projections. So like those neurons that are more in the cortex that's responsible for decision making and more conscious thoughts, they can actually send projections down to the AGRP neurons and tell the AGRP neurons to stop firing a little bit. So when an AGRP neuron is active, an animal's hungry, but we have some control over our AGRP neuron activity. Not in the way, I'm not saying it's easy to just tell yourself, okay, I choose not to be hungry now, so I'm going to turn it off. But we can override that in some deliberate way. For example, if we know that we're not in a situation to eat, we can focus on other things and get past that. So right, so basically there are mechanisms of top-down suppressing hunger depending on our environmental context and what we choose to do. Did I answer the question? I hope that was a meaningful answer.
Yes, I asked the question. I think it was answered. Yes.
Oh,
Thank
thanks,
you.
Marissa. Thank you.
I'm seeing Laura had a question. Laura, if you want to speak to your question, or I can read it again. I think I'll read it. She's kind of hoping for a simple list to share with clients of the factors that affect sleep. Oh, here you are.
Oh, yeah. Yeah.
I'm thinking to use therapeutically.
Yeah.
I'm not sure I have any clients who don't have sleep problems at this point. And when we start exploring the possible reasons, and I would say the way you explained it, the obstacles, that what competes with good, healthy sleep, so many of them may be items that the clients aren't even aware of. So I wonder if there is a way to explore it in a hierarchical sense, like that, you know, if you're really hungry, it doesn't matter how unstressed you are, you're not going to sleep, you know.
Right.
Or if you have a test tomorrow, it doesn't matter if you eat before you go to sleep, you're not going to sleep, like that.
Right. No, that's a really good question. So I think that no matter what, I totally get the question of a hierarchy. I think it's not necessarily that one outcompetes the other. So it's not like hunger is greater than thirst, which is greater than stress. They're all bad. And I think it's just more, what do we have control over? So hopefully if somebody's thirsty, they have access to water so they can get sleep. And hunger is something, hopefully if somebody's hungry, that they could make a choice to have a you know, something with maybe some protein in it or something like that. Stress is the hard one, because stress is something where, you know, if you're stressed about something, it's much more difficult to just turn off the stress. You can't just ingest something to turn the stress off. So I think that when I talk to people who have difficulty falling asleep, hunger and thirst don't tend to be the dominant reasons. Actually, stress is the number one reason why the college students I work with, you know, they're very stressed out about exams and papers and things that they have going on in their lives, and other emotional issues as well. And so we work through different ways of trying to alleviate stress at night, you know, there's obviously short-term stress reductions for a given moment versus more longer-term things that people can do to try to reduce stress in their lives. I don't know if this is exactly, maybe this is a tangent to your question, but I would actually say that the number one reason why people have a difficult time falling asleep and staying asleep, besides those kind of survival factors, is screens. It's just a fact that looking at phones and tablets and computers, that keeps people up and stressed more than anything else. And there's a neurobiological reason for that as well. There's a couple, actually. I could just mention really briefly, is that the circadian system that tries to get you to fall asleep at night and be awake during the day is all regulated by light. And so the amount of light that hits your eyeball when you're in situations of light, that keeps you awake. And, you know, throughout most of evolution, when the sun went down, that was it. There was no light, and campfire light and the occasional light bulb that is not so big. But now, you know, I have my phone on me right here. So imagine, you know, people now fall asleep like this, that it's the last thing they see before they go to sleep, and they're blasting light into their eyeballs. And I know that Apple and Samsung phones, Android, they have settings where you can turn down certain frequencies of light to make it a little bit optimal, but even that's not perfect. I mean, all the light that's hitting your eyes keeps you awake. The other thing is that phones are designed to stimulate your dopamine system, and everybody, you know, when people are on their phones, they often are on things where you flick to get more information. It's like you flick for new emails or YouTube videos or Instagram posts or something like that. And people have shown that every time you take your finger and you flick upwards, you're releasing a burst of dopamine into your brain, and the dopamine keeps you awake. So every time, that's what dopamine does. One of the purposes of the brain dopamine system is to motivate you to keep doing something, whether it's rewarding or engaging in any kind of way. And in humans, not just mice, but in human studies, anytime somebody is looking at Facebook or Twitter or whatever, and they flick upwards, they get a little burst of dopamine. And so that's the number one thing I tell students now is that I think this is the biggest difference between people now and 10 years ago. Is 10 years ago, we still were stressed and we were still, there were still these factors, but now everyone is like illuminating their eyeballs with light and constantly flicking. And the phone is what almost everybody says is the last thing they do before they go to bed. It's just like a lot of times people fall asleep to their phone, or they fall asleep on their couch flicking phones, or they, even before they turn off their light, or even if they do turn off their light, they're still looking at a phone or a tablet or something. So that's the number one thing I would say.
I just have to say that we have in this group learned about circadian rhythms, also learned about dopamine. However,
great.
The flicking is huge. Okay, I know it's the scrolling and it's just and I'm so happy that we're still recording, because just what you just said is, you are an expert. And people will have more confidence because this information is coming from you. But that is amazing. The flicking thing is
it's absolutely amazing. And the maybe one other thing I can say about it is that all of the tech companies know exactly what it is that they're doing. And reports have come out that, people who make these apps, they're designed so that you don't stop using them. Like they're designed, like the people who make, Instagram and all the social media apps, they're designed so that you don't want to stop using it. And so that's why, imagine every time you were on a social media app, you had to press a button that took you to a new page every single time. That would just require constant going from one page to another page to another page. So instead, they design it so that it's all there, and all you got to do is kind of swipe upwards. It's like you never have to leave to go to a new page. You just kind of see what's below that, and you're constantly driven. If you see something and it's not stimulating you, you think, oh well, I'm just gonna swipe upwards for the possibility that maybe then I will see some cool post by somebody who I know, or that will be stimulating. And that constant drug reinforcement of dopamine bursting is it's very well documented. So I'm glad that that's helpful.
Thank you. Yeah, I'm seeing Melissa had a question after that. Do you want to share what you had asked, Melissa?
Sure. Yeah, you kind of covered it a little bit. It was this idea of, you talked about quantity versus quality of sleep, but I would, again, if you're eating junk food, like chips and stuff, and especially doing it right before bed, you may feel completely full, but is that really giving your body the nutrients that will interact with the neurons that are defining hunger versus fullness? And so that was, but also we did learn about lizard brain snacks on the anxiety part of thing, the combination of eating some protein with a little like something that like fruit that might have a little to within, maybe 15 or 20 minutes or so, bring down the level of anxiety. So it's another food connection.
Great.
Of a factor before you go to bed. If you've got a test the next day, kind of, putting together combination solutions for dealing with the body and the brain connection, and what each one needs specifically, rather than just these generic, food, right, type.
Right.
Yeah.
Right. Even with carbohydrates too, I could say like having an orange is obviously very different than having a Snickers bar. They both have sugars in them, but the sugars in an orange are different than the sugars in a candy bar. And fruit actually also has a lot of other nutrients that go with the sugars that help you digest them. I think having an apple or an orange or something like that is the nutrients are in a really nicely proportioned natural package with fiber and other nutrients as well, as opposed to the density with which you get the sugars in something else that's more, like ice cream or something like that. And so a piece of fruit is great, actually. That can be really nice. Yeah, like
Yeah,
some protein.
I actually have over the last year, I was able to lose 80 pounds because I went on a healthy eating plan of 4 ounces of protein, 4 ounces of veggies, 4 ounces of fruit, with intermittent fasting, meaning after 8 o'clock at night, I don't eat for 12 hours, and
Yeah.
it made a world of difference to my ability, as you were saying, to stay asleep, fall asleep, have good quality sleep. And then that helps you through the day. So it's just one of those things, once you start spiraling all the positive effects that it can have, just recognizing these the ability of how your body even processes hunger. Because if you're not hungry because you're getting the right nutrients, then you're going to sleep better and so forth.
Yeah.
So, yeah.
Absolutely. There's a quick book. This is just a quick book recommendation that I personally like. So I'm just one person, but I like this book. But it's called The Obesity Code by Dr. I think his name is Jason Fung. And Jason Fung is a diabetes doctor, but he wrote a book called The Obesity Code, which maybe I actually don't even know that I like that title very much. It reminds me of the Da Vinci Code. It sounds a little gimmicky or something. But that book was all about how it just blew me away that the composition of the nutrients that we eat actually really matters a lot. That like eating something, again, like an orange, it's like we're not just having the sugars from the orange, but we're having there are nutrients in the orange that help you absorb the sugars differently over time than if you just had the sugars by themselves. And so the way the digestive system gets these nutrients and then experiences them together in natural foods, it actually really makes a difference which nutrients are combined with which nutrients. And in natural foods like fruits and vegetables, they're naturally packaged together in a way that's not packaged by something that like a microwave food that you get from a convenience store. It's like they might have the same kind of nutrients there, but they're not composed the same way, and it really matters a huge deal. That book was really interesting to me. So I think The Obesity Code about nutrition. But
Thank you. Yes.
I think Marissa made a point about that earlier. I think it was you, Marissa, about poverty and obesity and maybe some of the like deeper effects of poverty and not having access to food, like high quality food.
right.
I was curious, as you were talking to Melissa, is there a recommended food you would say to eat before bed if you're suddenly hungry, which is me?
Oh, that's a good question. That is a good question. I tend not to, I am a human being just like everybody else. I actually try not to eat a couple hours before I go to bed. I try to basically have a good dinner. And, since I've become aware of these things, I try not to look at a phone or a computer about 30 minutes before I go to sleep. I just say, like, you know what, I'm going to declare myself done with Facebook for the day, or whatever it is I'm looking at, email for the day, and I stop looking at it. But if I do get hungry, yeah, like, a piece of fruit, or sometimes if there's like a protein bar or something. I've been a fan of these natural, I don't even remember the brand, but they're little pepperoni sticks, except they're made out of chicken. It's like a little chicken pepperoni stick, but it's mostly protein. I think they're higher in sodium than I would like, but it kind of just naturally makes me feel a little full, and it doesn't have a lot of sugar in it. And that's what I like personally. But yeah, that's a great question. I saw there's a question in the chat here too about if there's diseases associated with AGRP function and metabolism, and there are genetic studies that show that people who have different composition, different genetic sequences for some of the genes that are both the agouti related protein, and then there are other genes in those neurons and nearby neurons that are also help control body weight. And there are natural alleles of these genes that make people susceptible to some and not the other. I don't know if people on the Zoom call are familiar with leptin as well. It's like leptin is a hormone secreted by adipose cells, by white adipose tissue. And the more adipose tissue somebody has, the more leptin that they make. And leptin has a huge influence on AGRP neurons and other circulating hormones as well. But there's not one magic bullet. I think, like, people have looked to see, is there one thing that it causes genetic susceptibility for something, and it turns out that it's usually much more complicated than that. There are some diseases where, there's a very easy to find mutation in a single gene or a single protein, and body weight regulation doesn't tend to have a single answer. There's multiple genes that have an impact. But the answer, the simple answer to the question is that there are genetic mutations and associations that play a role, not just with AGRP, but with other peptides as well, and other hormones like leptin.
Thank you. I also want to acknowledge your time and check in to see if you have time to stick around anymore. Or do you need to get going?
Sure. I'd be happy to answer a few more for sure. Yeah.
Okay. Yeah.
If people want to stick around, I also want to be sensitive to if other people would like to end.
Well, we usually
I'd be happy to do a
1:30.
couple more.
Yeah.
Okay. Oh, okay. Yeah, I'm happy to.
Sarah asked a question. Do you want to speak to that question about delayed hunger in the morning?
Oh, I
Right.
see.
In other words, when people wake up in the morning, and Eric, I'm one of those people, that I don't want to eat breakfast for at least an hour or so. And I was just
Right.
wondering, is that a function of AGRP dysregulation, or are there other neurons at play?
Yeah, that's a great question. The short answer is, well, I have two very immediate answers. The one immediate answer is that nobody knows about AGRP neurons specifically, and that's because it's really difficult to measure this in a human. In a mouse, this is why I use mice, is I can do all the kind of manipulations and measurements that I showed you in a very precise way. Right now, it's currently impossible to measure AGRP neuron activity in a human. We know that AGRP neurons are there, and there's some pharmacological things that we can do with humans, but nobody knows on a moment-to-moment basis how AGRP neurons in a human, as opposed to a mouse, functions. But the other thing, immediate answer I can think off the top of my head is I myself am never hungry in the mornings. I just I wake up and I was always told that breakfast was the most important meal of the day, and I just I finally, when I reached adulthood, declared that I will not eat breakfast. I just don't. To me, I'm not hungry in the mornings, and I don't think it means anything is wrong. There are some people who have kind of a different circadian schedule than others, and I am really fascinated when I see hormone level measurements over the course of a day for a typical person. I think there are some hormones in your bloodstream that peak at different times over the 24-hour period, and some peak while you're sleeping, and some peak in the morning, and some peak in the evening. But there's variability with all of those hormone peaks. And I think that the idea that there are night people and morning people is totally true, that there really is such a thing as a night person who is kind of more alert, and their physiology is a little bit more active at night. And then there are morning people who are just more engaged in the morning, and it has to do with these different fluctuations in hormones. And I just know that I myself am more of a night person, and in the morning I'm just not hungry. And I feel that that's completely natural. So there's really nothing to worry about if somebody is not hungry in the morning. Presumably my hunger kind of peaks around lunchtime. So I make up for it with a good lunch. That's what I would say.
It's interesting. I too am a night person.
Yeah, it's completely scientifically valid. Like, I mean, like calling people night people and morning people is true. There are night people and morning people. It's not just a common layperson's term for it. It just, and it has everything to do with peaks of hormones. It's just normally before people wake up, we get a burst of cortisol in our blood, the stress hormone, and it's not necessarily meaning that we're stressed in terms of anxiety. It just the kind of the increase in cortisol when a person wakes up helps prepare us for the actions of the day and helps make us more alert and more physiologically responsive. And some people get that burst of cortisol, you know, early in the morning, and some people get it later in the morning. And if the later you tend to get it, the more you tend not to be a morning person. A morning person gets their burst a little earlier than everyone else, and that kind of is what makes us you know, alert and fresh. And it's completely real, and that variation is very well documented and very real. So I've embraced the fact I was a night person. I think when I was in high school and in college, I felt lazy because I didn't want to work in the morning, and I, you know, did my best thinking at night. And now I've come to embrace that about myself. And of course, in the workday, of course, we have to go to work in the morning, and there are professional reasons, you know, we have to do work in the morning. But I've realized I tend to prioritize the things I need to be most creative about and most thoughtful about in the times of the day where I'm most alert. And I think once people realize that about themselves, then they can be more productive and more. But I agree with you.
Thank you.
Sophie was asking about your work with athletes. Sophie, you want to share some more about that?
Sure.
Oh, sure.
Yeah, I
Oh, yeah,
I
go
know
ahead,
athletes
Felicia.
need more food, and you mentioned working with athletes. So I was curious what you had to share.
Yeah, no, that's good. Yeah, no, with athletes, it's I mean, everything I tell athletes is basically a lot of it comes to just performance. I mean, again, this is something that maybe just seems obvious, and yet it's amazing how people really benefit from hearing it. But just how much people improve when they try to get enough sleep is really staggering. And one of the things that I've learned, I'm not a professional athlete myself, but I've learned that professional sports teams often hire sleep consultants to work with the players on their teams to get enough sleep and to try to force them to get enough sleep. And there have been famous studies that have been done, one notably at Stanford University, where at Stanford University, there was a sleep study where there were some teams that they've forced their athletes to get enough sleep, and by forcing them meant like they would get compensated for a study if they came in and they, you know, put on a sleep measurement device, and as long as they were getting enough sleep, the athletes were compensated. But then their performance went way up compared to people who didn't participate in the study and it wasn't just a little bit, it was a lot. And, you know, in these sports, especially when every second counts, you know, the difference between coming in first and fourth is half a second or something like that. It's just amazing that the people who get the most sleep tend to be the ones who benefit that much. And so I found that the student athletes at Williams College, where I am, they kind of know. I mean, everybody knows that they should get a good night's sleep. They, you know, this is something that gets tossed about, but I don't think that they really just see the physical, tangible benefits. And so when I find graphs that basically showed like, okay, here's an experimental group of athletes that were forced to get enough sleep, and here's the control group. And you can see the difference in sports performance. And they see that and they're like, oh, which group do I want to be in? And then they can force themselves to do that. I think at least the student athletes that I know are pretty good about nutrition. Like somehow nutrition has broken through. That they know that if they're, you know, performing the next day, that they shouldn't eat really bad junk food all week. And somehow they've intuited that, but the amount of sleep they get, they haven't intuited at a level that affects their behavior until they see graphs like that, and that really makes a difference.
Interesting.
Yeah.
Dr. Carter, I was thinking of your comment earlier about like, we might not be able to control all of those drives, but let's get our basic physio together, you know, and increase our sleep, thirst, hunger, all that stuff. And as we said, there's complications related to sociological issues that might make that hard for some people.
That's
But
right.
as counselors, when we're thinking about the transition, sometimes we kind of forget about the basic systems and like connecting people to food security programs and things like that. So I think it's just a good reminder of like, how do we help maybe from a case management perspective, the linkage to resource perspective. But I'm also like mindful, like there's a lot of sort of weight management medications, hunger suppression medications, medications that are designed for other things that are being used for weight loss. Like, I guess, what are your thoughts as how like these neuron manipulations could be used in maybe those efforts in humans in the future?
Yeah, so like in terms of the obesity drugs and
yeah, or even drugs that aren't for obesity that people will take to suppress hunger and things like that. Because you mentioned like, so optogenetics, if we were to stimulate the neurons that suppress hunger,
Right.
like, or turn those neurons off to suppress hunger, I don't know. I imagine there's some risks and benefits to those considerations, but I would imagine people are trying. So I'm just curious your thoughts.
That's a great question. I think I guess the first thought off my head too is just that the side effects of a lot of these medications that people take, whether they're over the counter or their prescription, I mean, not only is there a lot of variability, but they can affect people in different ways. I've personally shied away from, I maybe experimented is the wrong word. That sounds more out of control. But like there have been times in my life where I've tried taking over-the-counter drugs for things, and I found that it kind of just messed with my homeostasis as an animal more than it helped in certain ways. And then there have been some medications I've had where it's caused sleepiness as a side effect, or it's interrupted hunger as a side effect. And obviously the benefits of the medication can outweigh those side effects. And so, and everybody's taking different things. And so it's just, I think just being cognizant of it or knowing, you know, what they are, has helped me at least. I don't know, during times where I've been prescribed something and I've realized that it's having an effect on my hunger or my appetite or my stress levels. I guess, I don't know if this is a good answer, but excuse me. It surprises me how sometimes people are not cognizant of their own physiological states. Maybe for better or worse, you know, it's I don't know if like in the 90s, there was a very popular book called Emotional Intelligence. I mean, it's still pretty popular, EQ. But one of the things I remember learning from that book was just that a lot of times, a lot of people don't even know the emotions that they're experiencing at the time they're experiencing it. You know, it's just like somebody might be angry and then not really fully appreciate the fact that they're angry about something. You know, it's not as simple as, you know, like the way we tend to think it. And I think how that applies to survival systems is somebody might be hangry. I don't know if you know, like we all know this term hangry. It's just like you're angry because you're hungry, and you might not fully appreciate that, you know, like, oh, I'm experiencing this mood I am right now because I'm hungry and I don't know it, or, you know, so some other factor with that. And I think that what I learned from the emotional intelligence book was not only trying to be more in touch with what emotions am I feeling at a moment to moment basis, even though it sounds so simple. It's like, of course I should know what emotion I'm experiencing at the time. But I think a lot of times we're just so driven during the day, it's hard to identify that, but also just knowing our own physiological state at the time, you know, just asking yourself, am I hungry right now? Am I thirsty? If I'm feeling anxiety right now, can I dissect that anxiety and pinpoint it to something? You know, can I assign my anxiety to some sort of space, and it's actually really interesting to me how modern day people have a difficult time defining that. So how this comes to your question is, I think a lot of times people are prescribed a drug, or they take a drug, or whether it's over the counter or something, whether it's something even as simple as like, ibuprofen, or something, much more targeted to a certain part of the body. And they're not even really attuned to the side effects of what it is. It's just like, I wish sometimes that there was it wasn't just a list of side effects, but it's like, here's a list of questions you can ask yourself about what you might be experiencing in your body as you go through these things. And yeah, it's an interesting question.
So do you see a world where there's an AGRP neuron inhibitor at some point in the near future?
Yeah, well, that's the dream. There's actually, it's funny. I don't know if you're all familiar with the drug like Ozempic, or there's now these great obesity drugs that are GLP-1 agonists that actually they affect the brainstem and not the area where the AGRPR is at. But that's been the one and only success story in decades of research. It's been actually kind of amazing how difficult it is to target very specific neurons in the brain. And if there was a way to target AGRP neurons in a person, oh my gosh, that would earn whoever figured that out $1 billion, if not more. It turns out it's in mice. This is the unfortunate thing, is in mice and rats, it has become very, very easy for scientists to perturb very specific populations of neurons. And I can do just about anything I want to an AGRP neuron in a mouse that's moving around freely. It's almost impossible to do that in a human today. And the techniques that work in rodents just aren't feasible to do in a human. And pharmacological drugs are really the easiest thing to do in a human. But actually, if you just think about it, how many pharmacological compounds, what are the great success stories? There's SSRIs, there's this new Ozempic drug, there's, and you all on this Zoom call probably know more than me, but they're not targeting very, very specific populations of neurons in the brain. They're targeting global things like serotonin. And actually, what I've just read, I don't know if you have seen this too, but just a year ago, there was a review study on SSRIs that basically made the point that they're not even probably doing what we all think they're doing. Like they might not even be affecting serotonin specifically, and they're called serotonin specific reuptake inhibitors. They might actually be acting on something completely different. And so it's or in different ways. So it's yeah, Melissa, I see you're
Yeah, just along those same lines. And again, it goes back to my comment before. If you're taking something like a antibiotic that's wiping out all of your gut bacteria,
right.
including good gut bacteria, again, how is that relationship between gut neurons, and neurons of your brain in terms of this whole idea of fullness, hunger, need, chemical need to restore brain functions and the ability for the brain to carry out normal operations such as good sleep, right? How that's
Right.
all affecting, and nobody's talking about that, so to speak, in terms of just what is that relationship and what is that doing to your gut, and does, probiotics, again, do what you're kind of simulating in your experiments there,
Right.
that alters, in a good way, your ability to stay asleep and have quality sleep, and so on and so forth. Mm-hmm.
That's a great point. The gut microbiome has just been such an interesting recent field of study too, just in terms of, for decades, people have known that there's a gut microbiome, but just the degree to which it affects physiology and health, and also mood and cognition and emotions and behavior. It's just amazing that people have actually done studies that, the microbiome affects things you would never expect it to affect. It's just, having a mouse model of autism, and transplanting the microbiome from wild type normal mice into the mouse model of autism affects the mouse and makes it more like the wild type mice in a way. And so, these haven't been reproduced in humans, these studies, but the mouse physiology is very similar to human physiology in a lot of ways. And like, why in the world would symptoms of neurodivergency be attributable to bacteria in the gut? I mean, it's an amazing development. So a lot of these things, like the foods you eat, I mean, they could be directly impacting neurons in the brain, like we talked about earlier, but they could also be impacting the flora in the gut that will then indirectly impact things in a different way. So it's all very complicated. I guess is a global
I
way
liked your term confounding factors, because that's a pretty good way of kind of just, again, there's all these stuff, so let's focus on at least a couple of things that we, like you had expressed, that we can control,
Right.
and that's probably the most important efforts we can make in our well-being, physically and mentally and emotionally and all that. So yeah, very good.
Yeah, thanks. Yeah, great. Oh yeah, I see sleep beds by by athletes. You know, like I've actually seen that some big companies like Google have actually installed what they call nap pods in their offices. They're just like, I don't know where, I've never been to Google's campus or anything like that, but I've seen that a lot of companies are actually putting in these little pods where you can kind of isolate yourself and then take a little nap, because they find that people are more productive if they've if they're you know, able to take a quick nap or something like that, and that it's better than forcing people to stay awake and things. And so it's like, I think, yeah, getting a good night of sleep, yeah, there's something to it more than just comfort.
We're getting close to 1:30 for me. I'm not sure I'm always bad at time translation, but the 30. So I don't think we'll have time to talk about this, but as you were talking, I couldn't help but keep thinking about people living with anorexia and how much that must impact like every other part of their life too. And all
Absolutely. Yeah, absolutely. I guess that's maybe another, I mean, following with the idea that everything is so complicated, it's just that I think things are often more complicated than just the primary way people think that they are, you know. And it's like something like anorexia, you know, it's like there's an obvious initial set of symptoms which are very prominent and very obvious, but the secondary and the tertiary effects of the etiology of what people are experiencing. And sleep is often the one that I think gets talked about the least. You know, I mean, I think that, you know, like there's a lot of obvious or diseases or conditions and pathologies that people know about that have a very obvious set of symptoms. And then there's a certain set of symptoms that people don't often talk about because they're not as prominent and they're not as obvious. But sleep is obviously a major disturbance that then will have symptoms beyond that by the sleep loss. And so yeah, just something to be aware of.
It's also refreshing from a neuroscientist who also looks at very, very minute, like microscopic components that contribute to behavior, to also think about the whole system and all the other contributing factors that are there too. So I think it's nice to have those little pieces of research, those little findings that can then translate into other systems of functioning that we're talking about. And I think it's a good reminder that little changes can impact a lot of things, and for the maybe better and the worse. And, you know, I think it's a reminder to us that we might not be able to change a lot of things, but maybe there are some really tangible impacts that we can make on the short term.
Yeah, definitely. One thing I could say too, maybe is a good note to end on as well, is just that I've learned over the years that it doesn't have to be all or none. You know, like somebody who's not getting enough sleep or somebody who has a poor diet doesn't have to suddenly say, okay, I'm going to change my entire diet, or, you know, I'm going to suddenly be really good at sleep hygiene, whereas I wasn't before. It can be sort of a gradual process. And one of my friends reminded me of this, of like I knew somebody who was trying to be vegetarian and realized it was really hard. And they were sort of like, well, I don't know whether to be vegetarian vegetarian or not vegetarian. And then, you know, a friend made a point. It's like, it doesn't have to be binary. You can eat less meat and still, you know, impact your diet or impact the environment. You know, it doesn't have to be all or none. And so I think that was a great reminder for sleep research and for nutrition also, that, you know, like saying somebody could, you know, everybody should get some more sleep doesn't mean you have to suddenly, you know, alter your lifestyle tremendously. It can be iterative, and it can be, you know, just a little bit more can make a big difference too. Well, thank you very much for inviting me to this. This seems like a wonderful group, and I was really happy to meet all of you. Thanks very much. And yeah, feel free to email. I met my email. I can put my email in the chat if anybody wanted to follow up with anything. But I'm just at the Williams College Biology homepage as well. Yeah, thank you very much for all of this. It was nice to meet and talk with you all. Thank you.
Thank you.
And have fun at the rest of your institute.
Oh, thank you. Thanks very much. Thanks so much.
Sasha, who we got next month?
We have Dr. Jonings finishing part 2, and he said he'll be focusing on divorce, parenting, and loss of a loved one. So he called that like the second stage of relationships.
All right, cool. Well, Dr. Carter, you're always welcome to pop in as a participant too. So
thank you.
Feel free to drop in, and I wish you all well as you transition into your Julys, and hope to see you all next time.